great to be able to give this lecture. So the aim of what I want to do today is to basically give you an overview of what we've been doing in the lab for about the last 2 years, where we got interested in CRISPR and we wanted to adapt it to our own particular needs. And so what I'm going to show you is a series of tools and experiments and assays that we've set up where we've been trying to adapt CRISPR to quite a specific but I think important problem of how we can knock out non-coding elements in the genome. So most of the people began using CRISPR to knock out protein coding genes, and our main research interest is non-coding elements, and we have come up with a way that we think is quite versatile and powerful for deleting almost any non-coding element in human cells. So all the work I'm going to show you is done by these people, by Estelle, who's a postdoc, by Karma, who's our lab manager, by Taisia, Carlos, and Nuria, who are 3 students who've all contributed to these projects I'm going to show you. And this nice photograph actually basically done by Didac in our lab encapsulates the whole philosophy of what we're trying to do, and I think it'll become quite clear what the meaning of that is. So in the talk today, I'm going to give you, first I'm going to explain the problem that we're trying to solve with particular reference to long non-coding RNAs, although I hope it'll be clear to you that the tools which I'm describing are not just specific for these particular RNAs. You can use these tools for many different types of elements which you might want to study. Then I'll talk to you about this vector we've created called DECO. In the second part, this knockout vector. Finally, I'll talk to you about this software called CRISPR, which we've also developed, which is basically the design software for DECO or for any other type of deletion method. In case I use 2 different words interchangeably, link RNA or long non-coding RNA, they both mean the same thing essentially. And please feel very free to put up your hand during the talk and interrupt me or ask me any questions or doubts which you might have. So I'll just give you a little bit of biology background first to understand the motivation of what we're trying to do. So we're interested in these things called long non-coding RNAs. We've been working on these for quite some time. And one of the big problems with working on these genes is that they're very hard to knock down and they're very hard to knock out. And so it's been a real challenge to understand their function because we haven't had very good tools for manipulating their expression. And this is obviously why we got so excited when CRISPR came along in the beginning. So long non-coding RNA, well first this is a messenger RNA which we're all familiar with. This thing has got it's spliced, it has a promoter, and of course what's important about these, and we'll come back again to that point, is they have an open reading frame which encodes protein. Long non-coding RNAs, for those of you who don't know, look very similar to a messenger RNA. Okay, in terms of their gene structure, they usually have a promoter, they're usually spliced, indeed they're usually capped and polyadenylated, but the key difference here is that we cannot identify any protein coding sequence in those genes, and so we think that they don't encode any protein, that they function as an RNA molecule. And this, of course, raises unique problems, technical problems, in trying to knock them out, either with CRISPR or with other methods. So in a single slide to describe what we think link RNAs do, we think that the link RNA here is shown in blue. We think that these things interact with all kinds of other biomolecules in the cell. They can hybridize to other RNA molecules, perhaps regulating their translation or their localization. There are many reports suggesting that they interact with genomic DNA through some kind of canonical or non-canonical base pairing, and we know that they interact with all kinds of proteins, either through structures or through sequence elements. And so this leads to the idea, which again I'll come back to, that link RNAs may have domains, and that it would be cool if we could actually start to knock out these domains and see what the effect is on their function. And so the really big motivation here is that in the last few years we've discovered so many of these link RNAs in our genome. Okay, so let's say around 2008, almost nobody had heard of these genes, and we only had catalogs of maybe a couple of thousand or a few hundred of them. And in the last few years, a number of groups worldwide have made really heavy efforts to start making catalogs of link RNAs, and even the most conservative estimates, as I show you here, have 16,000 of these in our genome. Okay, compare this to just under 20,000 for protein coding genes, and you'll see that there's really a massive amount of novel genes here which we want to start knocking out and exploring their function. The other important thing is that of these 16,000 link RNAs, we know the function of probably less than 1% of these things. Okay, so 99% remain completely unprobed, and we have a big challenge in trying to screen and understand the function of these genes in an efficient way. And so the questions which are driving our research and why we're trying to develop these CRISPR tools, as I said, what is the function of individual link RNAs? If we do an experiment, we see a change in expression of a particular gene, can we knock out that particular gene and see what the effect is? Can we start to look at the level of domains of these link RNAs and start to delete individual pieces of these genes in the context of the cell and see if that affects their function, and try to break up these genes into their constituent parts? And really what the ultimate aim would be is, can we not only look at individual link RNAs, but can we start to screen thousands or all of them in a single experiment and knock them all out in parallel and find the ones which give a particular phenotype? And what, of course, I'm going to tell you is that we think that this CRISPR-Cas9 may be the tool that can do all of these things. So I guess you've seen this type of figure about 20 times in the last 2 days or something, but I just wanted to clarify that in the talk today, I'm going to talk about natural Cas9, so none of these inactive mutants, none of these fusion proteins. We're just working with the actual regular Cas9, which is the nuclease. And when we talk about the 20-mer recognition sequence of the short guide RNA, the single guide RNA, we call it the protospacer. So the protospacer, I think, is generally recognized to refer to these 20 nucleotides, the 20 nucleotides which you're designing when you're actually choosing your target site of the genome to cut. So I think that's all I'm going to say. They've seen this lots of times, right? I guess this whole thing. And of course, we expect, although it's not always the case, as I'll show you, that the double-stranded DNA break occurs about 3 nucleotides upstream of the PAM sequence. So what Mark was talking about yesterday, the more conventional use, or the first uses of CRISPR, were applied to disrupting protein coding genes. So this is actually very powerful and very straightforward, because it's very easy to disrupt a protein coding gene. It's very easy to disrupt an open reading frame, because all you have to do is to recruit your Cas9 to a more or less 5' region of the open reading frame. You get an indel, and the indel either introduces a stop codon or it introduces a frame shift. And in the majority of cases, the entire downstream peptide sequence is completely wrong, and you've essentially knocked out your protein. Okay? So all you have to do in these cases is to use a single guide RNA. In, I think, the latest reports is that not exactly the most 5' region, but somewhere in the first 5' half of the open reading frame is sufficient to get extremely high knockout of all protein levels. But in our research, we think, well, we have non-coding elements, okay? So these things don't have an open reading frame. How are we going to do this? It's not enough in most cases to introduce a single indel. If you have an enhancer or other type of element, probably one nucleotide change or an indel or something may not really completely knock out the function. In fact, it may not affect the function at all. So for things like link RNAs, we think that most of the sequence of these genes is kind of linker sequence between small regions of important nucleotides. And if you start introducing indels in those parts, you probably won't affect the function at all. So we have to think of a different way. And of course, it's not just us looking at long non-coding RNAs. There's all types of research groups looking at different types of non-coding elements, which there's a lot of interest in knocking out. You've got enhancers, insulators, microRNAs. I mean, this is one of the powers of CRISPR, is that whereas in the past people were using short hairpins, could only really start to screen protein coding genes, CRISPR allows us to really start accessing all these other interesting parts of the genome that we could never really touch before with the available tools. And as I said, in all these cases, or most of these cases, a single indel from a single guide RNA will not be enough to knock out the function. And so, other people, we didn't come up with this idea, but other people started playing with the idea of using 2 guide RNAs in order to completely delete a piece of DNA. So the rationale here is that instead of using 1, you use 2, and that these are on sites flanking the region which you want to delete. And then if you're lucky, you get your double-stranded breaks simultaneously, and then the cell will repair the break using the non-homologous end joining pathway, and you have essentially deleted the target element. Okay, so it's extremely simple. The only difference is you're designing two guide RNAs flanking your target element. So as Mark said before, I'm not going to dwell on this, but after you introduce a double-stranded break, there are two competing cellular pathways for repairing this damage. There's the homologous recombination, homology-directed repair, and then there's a non-homologous end joining. Okay, and as far as I understand, this is the dominant pathway, and this is the pathway which we're interested in here. And so the more specific objectives of our research has been these things. So firstly, we want to be able to try to delete or actually silence link RNA genes. We want to try to delete pieces of those genes without interfering with the expression of the whole gene. We want to see if we can delete multiple genes in a single sample of cells. And some qualities that we want these tools to have, we want them to have high activity and obviously high specificity. Although actually, I have to say that this question of off-targets and on-targets, I think for our type of experiments, is not one of the key driving forces. In our type of experiments, we don't care so much if we get some off-target effects, as long as we can get some reasonably high efficiency of deletion of the things which we're studying. And we've also put quite a lot of effort into developing some methods to actually quantify the efficiency of this deletion. In an analogous way to the surveyor method, we've been working on some simple ways to actually delete something and check how efficiently we're able to delete it in a sample. Okay, so that's the kind of justification and to give you some background on what we're doing. And now I'm going to talk firstly about our vector system called DECO. So, as I said, a lot of people have previously come up with this idea of introducing two different vectors or two different guide RNAs in order to delete a target gene. And it's very obvious that there are two different ways you could go about this. You could either take your cells and make two different vectors, each expressing one guide RNA, and co-transfect them. Or you can put both of your guide RNAs on a single vector and just transfect that vector. Okay, and you might think, well, each has advantages and disadvantages. And I think it'll maybe become clear to you later on what the advantage of using a single vector is. Anyway, we made this adapted lentiviral vector, which we call pDeco. It has the usual stuff. It's got a fluorescent marker. It's got puromycin resistance. But the important part is it's expressing these two guide RNA genes here. Each of the guides has a different promoter element in order to stop interference or prevent interference between the two promoters, which can be a problem, but they're both very high strength promoters. And what you should pay attention to is that the two guide RNAs have a variable region, the protospacer 20 nucleotides, which is the region which you will have to design for your particular target. And then you've got this scaffold region, which is the structured part that the Cas9 recognizes. And that structured region does not vary between any of your experiments. Okay, so the important point is that you've got these blue regions here and here, which will be different for every experiment you do. And they're separated by these constant, the gray and the orange here, which will always be the same. Some other important features, so it's quite versatile because it's a lentiviral vector, so you can either transfect it or you can make a virus and use it delivered by infection of your cells. You've got nice selection markers, you've got different promoters, which is quite important, I think. And as I'll show you, the cloning method for preparing your vectors is quite useful and convenient. So yeah, the cloning. So I hope I can make myself clear. And if anyone has doubts after I explain this, please tell me, because maybe you don't have so much background. It's a little bit complicated, but it's quite important. So we developed this two-step cloning method to create these vectors, okay? And essentially, the first step is you open up your destination vector and you synthesize or you order an oligo of 165 nucleotides that contains all of the variable sequence for your experiment. It contains both of these variable 20 nucleotide targeting sequences in one single oligo, right? But it's impossible to synthesize an oligo long enough from commercial vendors. It's impossible to synthesize this whole piece, okay, unless you pay an absolute fortune for it. So what we do is we synthesize this fragment, okay, but this fragment is actually lacking a piece in the middle. And so then what we have to do is, after putting this into the vector, then we open up through 2 other restriction sites in the middle, and we just pop in this part which doesn't vary between your experiments. Okay, so this is why we call it this 2-step cloning. We don't use regular ligation cloning. We use this Gibson method, which is a much more rapid and efficient way of cloning that you can read about. Many people are adopting this Gibson assembly. And by doing it this way, which may be a little bit counterintuitive, it gives you a lot of power and versatility for scaling up your experiments to very large scales. And I think it actually doesn't represent much more complication over having to sequentially introduce first this variable sequence and then second that variable sequence, which is what people are doing now. Okay. So I'm not going to explain right away why we did it like this, but it'll come in about 20 minutes, and I think it'll be obvious why we adopt this strategy. Okay, so now that we clone these constructs, we can basically design these 2 variable regions. We can select any target in the genome that we want to delete, and then you've basically got 2 choices about what type of project you're going to do. You can either, as I'll show you, derive a clonal cell line where you've basically got a cell line where all of the cells are homogeneous. They've all got the correct mutation, and then you can use that as your tool for studying your particular gene or your particular biological process. In the second approach, you can actually just infect your lentiviral vector and work with a mixture of cells where some of the cells will be homozygous knockouts, other cells will be heterozygous, and other cells will not be knocked out at all. Okay? And it kind of depends on what type of project you're working on. If you wanted to derive a clonal cell line, the advantage is you can get 100% pure cells if the experiment works, and you've got those cells frozen down and you can use them forever. So the cells are isogenic. The disadvantages is that you go a bit crazy at the end because you have to sort out sometimes hundreds of individual cells from a fax machine, grow them up, and genotype them, every single one, until you find a cell that has your correct knockout. And this crucially depends on the efficiency of this knockout, and the efficiency is sometimes not as good as we'd like it to be. So people in my lab who've done this will attest to going a little bit nuts, I think, after having to go through this process. But it has rewards in the end. If you work with what we call bulk cells, a cell mixture, you infect with your deco vector, and then you've got a mixture of cells where all kinds of things have happened. And again, the disadvantages, the advantage is it's quick, it's easy. You don't have to do this selection of individual clones, but now you're working with a mixture, and it just depends how important it is for you to have knocked out your gene in every single cell. If the effect that you expect to see is large enough that it's fine for you to just delete half of the copies in this mixture of cells, then that's fine, and you'll save a lot of time.
What is the efficiency to achieve 100% homozygous knockout?
Here, yeah. Well, okay, so the efficiency, as I show you, is about 50%. But what we're talking about here is that you actually infect with deco, you wait a little while, okay, and you select individual cells and you grow up those cells. So within one well, if you're lucky, every cell will be a 100% knockout.
And out of this 96 well plate where you have faxed your cells, so how many clones would be?
Right, well, Estelle, who did the experiment, is right behind you, and it's about?
I would say 2%. 2%. I would say it depends on also what are you targeting, so it will depend on your guide RNA. So at the end it was between 2 to 5%, and this is what people in the we are seeing also. So complete the deletion.
So what should we expect that half of the plate would be?
No.
No.
And also because not in all wells will grow cells, so low efficiency, but it works.
Yeah.
Which steps did you use the system so far?
Heck are the best, I think. Yeah. Yeah. Well, another very important point is that there are many cell types where you cannot grow individual clones, and this is also a problem. And so, we're still at early days. We're thinking about tricks you could do to grow a clone, with other cells, or or methods to improve the efficiency in the first place to boost the percent, but everyone has the same problem.
Thank you.
Right, so I'm gonna show you some experiments we did with this first part, which are published. So this is coming back to what I say. So this is the kind of workflow when you're trying to derive an individual clone, how it looks. So to give you an idea of the time frame, you're going to take about a week to design and clone your deco constructs. You have to do some infection or transfection. You got your fax, and then you start having to wait. You fax sort all of your individual cells, okay, into individual wells in a 96 well plate or 384 well plate, and then you just simply have to wait. Let these things grow up from an individual cell until you have enough to start doing genotyping. And then you start genotyping all the cells and you look at whether they're a knockout or not. So it's not necessarily, well, it's labor intensive, but the main thing is the time it takes. It's very slow. So when I talk about genotyping, what do I mean? So we do PCR-based genotyping, which is quite simple. So if you look down here, imagine this is the target region you're cutting. We just play around with different primer combinations, basically, to look inside our cells and see which alleles we have in that cell population. Remember that cells have at least 2 copies of everything, and these horrible cancer cells we use often have 3 or more. Okay, so when I talk about heterozygous, it could be 1 mutated allele or 2 and 1 wild type, or it can be a mixture. So if you look first at this panel, okay, so we have primers outside our target region, and basically if this is deleted, you get a short PCR product, and if it's wild type, you expect to get a longer product. And you can see in this first column that we have at least 1 deleted allele in that mixture, and in these ones we see the full wild type allele. Okay, you see the longer product. And then what we have to do, because of this problem of having multiple alleles in the same cell, we have to use another primer combination where now we put a primer inside the deleted region. So this should only amplify wild type alleles. And you see now that we still get a band in this red. So this red H is the same cell sample between these two. So that means that these must be heterozygous cells. They must have at least 1 copy of the wild type and 1 copy of the mutated allele.
Why didn't you get a second band then over there on the left?
Here?
Yeah.
That's a good question, actually, because I think what happens is that the shorter product always outcompetes the longer product.
Yeah.
So you have to be very careful. Just because there's no band here does not mean that there's no wild type alleles. Yeah.
Did you do the PCR on the genomic or on the cells directly?
Another good question. So in the beginning, we used to purify the genomic DNA, and then we realized that you can just actually take an aliquot of cells and almost, you put it directly in, right, Estelle, or you lyse it first?
With an acid lysis solution. I mean, you just lyse it with a buffer. In the paper, it's written the buffer composition. You just boil them and make them explode, and then the PCR that lyse it.
Yeah.
Really fast.
And both are efficient, or one is better than the other one?
For most of them, both are efficient. What we couldn't, in this lysate, in this fast lysate, we couldn't do the genomic PCR, we couldn't do these genetic PCRs for very long amplicons. So when we were deleting 3 kb, we couldn't make it to do this kind of PCRs with these lysates. But for regions that we deleted till 1 kb, it was fine.
Estelle should be giving this lecture instead of me. So another interesting thing that we found, which was kind of unexpected to us, but we had a lot of unexplained experiments until we realized that this was happening, was that instead of deleting, sometimes, quite frequently what happens is that instead of deleting this fragment, it actually flips it around. Okay, so you get an inversion, and it happens surprisingly in a very large number of cases. And we tested this, then we had all these strange results, and we spent ages trying to work out why these things were turning out the way they were. And then we realized, and then you stick a primer, 2 primers pointing like this, and then you start to see a band in some of the cell samples. So what's happened is in particular clones, you've had this switch, and you see the phenotypic effects that you expect from a deletion, but you still, when you do the PCR with the 2 primers outside, you still see a long product, and that's the reason. And so we see this a lot of the time.
Have you seen duplications of the fragment?
I don't think we've seen that. No. We've never seen a band longer in one clone than in others, so no.
But most of the times this would give you already a kind of like a knockout phenotype, right?
Right,
Because
right. Well, it depends what you're looking at. Because the things we're deleting, it's fine. But maybe if you're doing an enhancer, if you flip an enhancer, maybe it doesn't really have much effect. Okay, so here's an example. So now we isolated some clonal cells. So here's a gene, the transferrin receptor gene, which is an essential protein-coding gene. And what we were trying to do is delete the promoter of it. Okay, so we're only cutting the promoter and asking then what is the effect on the gene expression that we see. So importantly, these are heterozygous cells. We isolated one clone which is heterozygous. We could not isolate a homozygous clone because then we realized that the gene is essential, so the cells will die. But what was nice in the heterozygous cells, actually the genotyping which I just showed you, if you do FACS on the wild type or the mutant cells, we see exactly half the level of protein as what we expected. And we also saw half the RNA. It was actually very precisely half. Another thing which you can do is you can take your PCR products and clone the individual PCR products using TA cloning and sequence them, and then you can actually look at the exact nature of the join of the deletion site, okay? Because contrary to at least what we've read in papers where they say this is an exact cut 3 nucleotides upstream, we don't see that in the majority of cases. We see all kinds of messy cutting going on. So here is wild type. So here's the 2 PAM sequences here and here. And the bars indicate where in the literature you would expect this 3 nucleotides upstream cutting position to be. And you can see that in 3 out of 4 cases, we don't get this cut. You often see really weird fragments of sequence that have found their way in, extra cuts, and so on. Here's an exact cutting position. So, okay, so we were really interested in link RNA, so I'll just show you some data now where we're trying to knock out a link RNA, a cancer-related link RNA called MALAT1, which is a very widely studied gene, which is extremely highly expressed and very difficult to knock down by conventional methods. MALAT1 is this green bar, okay, so it's being transcribed. This is a genome browser shot. This is like a 2 kilobase marker bar here. The gene has 2 promoters here and here. And what we did is we designed pairs of guide RNAs which are marked by these colored bars, where the ends of the bars indicate the position of the guides. So we tried knocking out this promoter, we tried this promoter, and then this orange one covers both the promoter elements. And we transfected these different constructs. We selected cells. We looked at the expression. We genotyped these cells. So here you can see the shorter band as you expected from the knockout alleles. We managed to derive some cell lines which were homozygous knockout for the promoter of MALAT1. And then we did PCR. So we designed all these different primer sets. I'll only show you one primer set, but they all agreed in the results. which you can see here. So these are a few different clones which were isolated, just looking at the RNA expression by QRT-PCR, and you can see that, you know, in the worst cases we get about a quarter of the wild type expression, and in one of the clones we got extremely, almost complete knockout of MALAT1 RNA. And then so MALAT1 is known to give a proliferation phenotype. When you knock it down in cells, they proliferate less rapidly. And we saw that with these cell lines which we derived. And the amount of, so this red bar corresponds to this one. So we think that the amount of reduction in growth correlates a bit with the reduction in expression of MALAT. Okay, so that was all the deriving clonal cells. Now I'll talk a bit about how we've done some experiments with then mixed populations of cells, which is the kind of more practical way for many experiments. So in this paradigm, which we call the bulk cell, the cell mixture approach, we simply transfect our construct into cell lines. I should say that these cells we work with are stably expressing Cas9. Okay, so we made stable cell lines. They all uniformly express high levels of Cas9, and it's only the guide RNAs which we're introducing. We culture the cells for a while, and then we select to remove the non-infected or non-transfected cells in the mixture. And then after a few days of selection, we simply directly extract the genomic DNA. So we're not doing any sorting, we're not trying to choose the homozygous knockout cells, nothing. So we're more interested here in looking at what efficiency the deco is actually giving us. So again, coming back to MALAT1, in this experiment we didn't try to delete the promoter anymore. We were interested in whether we could try to knock out a fragment of the exon. So this is coming back to what I was saying, can we actually just delete pieces of a gene or pieces of a link RNA rather than silence the whole thing. And we also did an enhancer, but the results were quite similar between the two. And so now we had a big problem because, you know, to assess the efficiency of regular CRISPR, you have the surveyor assay, which is an assay for indels. But for these deletions, there was nothing really published that we could find where people looked at the efficiency of making a deletion. And so we came up with this very simple thing, which we call the QCPCR, a quantitative CRISPR PCR. It almost doesn't deserve an extra name. But this thing works by, you basically put primers, one primer inside your target region and one outside. And so this will only amplify wild-type alleles. And then you normalize it to a reference region, which is far away, just to control for your DNA levels. So essentially what this is doing is measuring the amount of non-mutated alleles in your mixture. And so firstly, we just checked that this thing was quantitative. So this was, Taisia did this. To check if it was quantitative, we need to make a dilution series of genomic DNA starting from, with known concentrations of a particular allele. And so to do this, what we did is we took those heterozygous cells, which I showed you, the TFRC knockout cells, and we took wild-type cells because we know that they're diploid. That's important. We know they're diploid. We know that they're heterozygous. And so we took the wild-type cells and the heterozygous cells, and we mixed them in different proportions. Okay, so here we have 100% wild-type, and here we have 100% heterozygous. So these have 100% wild-type allele that we're targeting, and these have only half. And then if you look at the yellow bars, this is basically the prediction from this QCPCR technique, and the gray is basically another primer set which is far away. Okay, so what you can see is that this QCPCR technique is more or less efficient and accurate in estimating what proportion of wild-type alleles you have in your mixture. And so now with this tool, we can start to go and look at the efficiency of deletion that we get. Okay, so here I'm showing you this exonic region, so where we designed these 4 different deco constructs, 4 different pairs of guide RNAs to try to delete this black region here from the exon of the gene. And again, as before, this is a control. The green shows the prediction from the QCPCR. The gray is again an unrelated primer set that's a control. And you can see that for these 3 different pairs, this pair is not so effective. For these 3 pairs, you get this reduction of about 50 or 60%. Okay. So what you have to remember, this is a reduction of 50 or so % in a pool of cells. And what we're measuring is alleles, individual alleles. We're not measuring homozygous cells or heterozygous cells. Okay. But we think that probably translates to about 20 or 25% of homozygous knockout cells, but we don't actually know that. And then, we also take these same cell types and we do this more conventional genotyping, which I showed you before, where you have your 2 primers outside and you can see these shorter bands indicating the presence of these knockout alleles. And so, coming back to this question of can we knock out a piece of a link RNA and leave the expression unaffected? So what Taisy did next is actually take these cell samples, and instead of looking at the genomic DNA to see if you've got the knockout, is to actually look at the RNA which is produced from that locus and see if the knockout is transmitted into the RNA, which is what we're really interested in. And so this was a simple RT-PCR, again, with these same primers flanking the deletion region. And this is with cDNA. And these are control cells, and you see you get the long band. And what's nice then is that in the knockout cells, you see the appearance of this short band in the cDNA. So not only are you deleting in the genomic DNA, but that's also getting into the RNA, which is exactly what you would want in these experiments. And you can also use the same approach with the TA cloning to look at the deletion sites. And again, none of them, or only one of them, matches what it's supposed to be. So it looks like it's quite promiscuous and non-specific, the exact position that the nuclease is attacking. Okay, so that's kind of the end of this section. Are there any questions about this stuff? No? Right. So next I'll talk briefly about how you can scale this. So another thing that we're really interested in is whether we can start using DECO to do big screens where we're actually knocking out hundreds or thousands of link RNAs in parallel. Instead of just choosing MALAT1 and working on it alone, we're trying to see if we can screen and use that as a way of pulling out interesting link RNAs from the whole population. So this is coming back to why we made this funky 2-step cloning method for DECO. It's precisely for this reason. So as I told you before, all of the variable sequence in DECO is in this first 165 nucleotide fragment. And the reason we chose it to be this size is because it's suitable for high throughput oligosynthesis. So I think Mark mentioned this yesterday. You can buy from companies, or you can do it yourself. You can actually synthesize very large, complex libraries of oligonucleotides. You can synthesize them on microarrays and order them in a tube as a pool, many thousands, tens of thousands of individual sequences all mixed together. And this is an amazing technology if you can couple it to things like DECO in order to perform high throughput screening. And happily, these companies offer synthesis beyond 165 nucleotides. And so what we can do is design big libraries of sequences and clone them directly into this backbone and introduce them into cells. And so to give you an idea of the type of experiments you can do, again, other people are doing these types of things. This type of technology was developed by people working with short hairpin RNAs over the last 10 years, but it's very readily adaptable to CRISPR. Here you take hundreds or thousands of your target elements, your target enhancers or microRNAs, whatever you want. For each of those 1,000 elements, you can design, say, 10 different pairs of guide RNAs. So that gives you a library of tens of thousands of different sequences. You synthesize that on a microarray, which is technologically very feasible, and then you can clone this into DECO. You can make a lentivirus from that and introduce that into your cells and do all kinds of screening experiments where you start to then do very efficient screening, pull out the cells which have a mutant phenotype and sequence those cells and find interesting new functional regulatory elements. Okay, so I'm nearly at the end. I'm just going to talk a little bit about some software that we did, so some design solutions. So I think you've seen a lot of bioinformatic design methods for designing optimal guide RNAs. And many people are doing this, and we made a software called CRISPR. In Catalan, it means popcorn. I don't know why, it sounded funny, right? It doesn't have any profound meaning. But we realized that, although there's so many different software packages for designing guide RNAs, there was nothing which was suitable for designing pairs, which was very surprising. And so that's what we did with my student Carlos. Basically, it's a problem not only designing pairs of guide RNAs, but how to design thousands of pairs of guide RNAs. So to put the problem more formally, basically you have your target region, again, your enhancer or whatever you want to study, and you want to just make large numbers of these pairs. Each of these is a protospacer, the 20 nucleotides, which is what you're going to clone into your DECO. And you have to somehow specify the regions upstream and downstream of the target where you want the software to design these pairs, some way to select the best candidate pairs. How are you going to choose the best scoring or some method through the distance between them? Anyway, Carlos, a master's student, wrote this software basically where you introduce, it's very user-friendly, you introduce a file specifying your target regions of the genome that you want to delete. So it can take one target region for people who are just studying one element, or it can take thousands. It doesn't care how many there are. And basically it does all this analysis. It defines the regions where to find the guides. It finds the best guides themselves. It starts assembling the guides into pairs, and then it chooses you the optimal pairs or ones with the optimal predicted activity and for your experiments. And again, it's adapted either for people doing individual experiments. There's a web server I'll show you that you can just design for your particular favorite region, or you can, through the web server, design entire libraries, or you can use the software from the command line and do it in a more bioinformatic way. This is just an example of the types of designs you have. So here's a protein coding gene. It's going on the antisense strand. So here's a design against the promoter of this gene. So you see these are all the different pairs that CRISPR has selected. One important thing, as you'll see here, is that many of these pairs start with the same single guide RNA. You see they're all together here. So basically the software finds, for whatever reason, that this guide is super high scoring, and it tries to pair that with many other pairs. And so one of the parameters in the software is deciding just how much a single half of the pair, a single example can dominate all of the pairs. So you can tell it, don't give me 100% all starting with the same single guide RNA. Anyways, just details. Here's another, an upstream element, an ultra-conserved regulatory element, and there's another 10 designed pairs for that thing. It works for 5 different model organisms at the moment, and if people really need something else, we can probably introduce it as well, but I think that these cover most of the experiments which are done presently with CRISPR. And the important thing is how effective it is. So effectiveness is basically in terms of if you're asking the software for 10 different pairs, in how many cases is it able to give you 10 pairs? Because often it cannot find enough guides within the parameters you give it. Okay, so here's a selection of different types of genomic elements which Carlos was running it on, and this is saying what % of these elements get as many designs as you ask for, how many get an intermediate number, and how many cannot get any single pairs. Okay, so actually this is quite bad for random. There's a lot of difference between what types of elements you try to delete. So if you take random regions of the genome, it's actually quite tough to design because they're very repetitive and they've got other properties which are not very conducive to designing guide RNAs. Whereas if you take, for example, protein coding genes, which are much less repetitive, 82% of the genes that Carlos gave it, it was able to give it a full 10 pairs for deletion. Anyway, I'll just quickly cover, I guess you know this already, but important aspects for these designs. You know, there are many or several different scoring algorithms available now that will predict the efficiency of guides, and they're all kind of competing and they all disagree to some extent on who's the best. A very important parameter is how much space you give the software to design your guides. It's kind of obvious. If you tell it, look in a window of 1 kilobase or 10 kilobases, it has much more chance of finding you a good guide RNA in 10 kilobases. Another important thing is this distance between the pairs. So we think that pairs which are very close together have much more efficiency than ones which are far apart, which is kind of obvious. And well, in the software you can tell it how many pairs you want for every gene and how strict it has to be with off-targeting, potential off-target effects of the different guides. And basically the last thing, so CRISPR is now available also as a web server. If anyone's doing these experiments, it's very user-friendly and you can control all of these different parameters I told you, and it gives you a report at the end with lots of useful information about the design and the guides which is designed for you. And actually what we're working on right now is adapting this for protein coding genes, because we think that although this system was designed for non-coding RNAs, if you actually design this system for protein coding genes where you're not targeting with one guide RNA, but you're targeting with 2 at the same time, it should improve the efficiency that you get in the knockout. Okay, so the time is nearly up. I just want to tell you this stuff, where you can get this from if you're interested in using it. So all the DECO stuff is published. The plasmids are in AdGene. It should be very easy to find. The CRISPR paper is on bioRxiv if anybody's interested in reading it. And I can give you the web server URL if anyone needs in their research to make those deletions. So just some acknowledgements. We did all this work in Roderick's lab over the other side of the building. These guys, as I said, did almost all the work that I showed you. Tony and Julia did the web server for us, and Carlo, as always, is a super great source of CRISPR information and also the reagents, and that's it. So thanks very much, and if you have any questions, please put your hand up.